Good morning to all Breath Biopsy Conference 2021 attendees. My contribution today will be focused on the potentialities of breath analysis for the early detection of oncologic diseases, highlighting the outcomes so far obtained, the limitations still to overcome, and the future challenges. Human breath analysis in terms of a chemical characterization of volatile organic compounds and the identification of a characteristic disease pattern is today recognized as a promising methodological approach, alternative to traditional diagnostic exams, for the early detection and follow-up of several oncologic diseases. Biopsy's composition in exhaled breath reflects the volatile composition of the bloodstream and airways, therefore is able to provide information on human metabolism. VOCs in the exhaled breath, to date estimated in over 3,000, can originate from two main sources: exogenous volatiles that are inhaled or absorbed through the skin and then exhaled by the subject, and those endogenously produced by different biochemical processes. Basic cellular functions, including energy metabolism and oxidative stress, are all known to be linked with VOC formation. Therefore, it's reasonable to assume that VOC's composition of the exhaled breath, that is the breath print of the individual subject in each respiratory act, depicts the cellular physiological status and then the overall health state. Another recognized strength of breath analysis with respect to conventional diagnostic investigations is that breath tests are simple, fast, safe for the patient, for the medical personnel, suited to all categories of subjects, and repeatable with high frequency over the time, allowing not only to early detect pathologies, but also to monitor potential metabolic disorders in subjects at risk, for instance, induced by pollutants exposure, or to monitor metabolic alterations in patients during the pharmacological treatment and in follow-up with patients after the surgery or the chemotherapy treatment. The potentialities of breath analysis in the identification of biomarkers for several oncologic diseases and also for specific cancer mutations have been exhaustively highlighted in the recent scientific literature. Over the last decades, the number of scientific publications has grown exponentially, with an increase only in the last decade from approximately 6,000 to 14,000. The promising outcomes to date obtained by the international scientific community are the result of further knowledge gained in the field and the progressive advancement of analytical technologies, along with the application of high-performance statistical tools. Despite the promising outcomes, breath analysis is still far from being applied as a routine clinical technique due to the lack of standardized procedures for sampling and analysis able to guarantee reliability and comparability of experimental results. Offline methodological approaches based on the collection of human breath inside polymeric bags or other inert material containers, the successive transfer onto adsorbent cartridges, and GC-MS analysis have been the most widely applied. Nowadays, we are progressively assisting to development of devices for the automatic collection of human breath and VOC adsorption of adsorbent tubes, allowing to skip in this way the intermediate step for the bags. This advancement is revealing more and more to be strategic in reducing the potential sources of simple contamination. Concurrently, online devices based on the direct collection of the exhaled breath and the response of a single sensor or a sensor array may provide complementary data able to increase the power of the statistical approaches for discrimination. Given this, the sampling procedure and modality of storage still remain debated and open issues within the scientific community, claiming for standardized procedures. Moreover, another critical point regards the factors playing a role in the definition of VOC's breath profile. The breath composition may vary from both a qualitative and quantitative point of view by subject to subject and within the same subject over the time, and is affected by exogenous VOCs of the ambient air where breath is sampled, the environmental exposure to VOCs, confounding factors such as sex, diet, genetics, and smoking habits, and differences in VOCs diffusion from blood to alveolar air, depending on their physicochemical properties. All these factors may interfere in the identification of disease markers in breath analysis. At this point, I'm going to discuss on the promising outcomes obtained by our research group over the years 2013-2020, in collaboration with oncology research institutions and leading technology partners. The first observational study is dated 2013 and was aimed at the identification of a diagnostic VOC spotter in patients affected by colorectal cancer. 78 subjects were overall enrolled in the clinical trial. More specifically, 37 patients affected by colorectal cancer, histologically confirmed, enrolled before the surgical procedure, and 41 healthy volunteers. After being collected inside the Tedlar bags, breath samples were transferred to adsorbent cartridges. The cartridges were then thermally desorbed and analyzed by GCMS. The entire methodology was set up and optimized, paying attention on each single phase of the analytical procedure and choosing the best available technologies at the moment. The study was designed in two subsequent phases. The trial phase was based on the chemical characterization of breath samples provided by the study population. This phase allowed to identify 58 VOCs of interest and to set up a VOC pattern for discrimination using multivariate statistical model, the probabilistic neural network. Based on the selection of the most significant variables, performance of the constructed predictive model was evaluated on blind samples in the validation phase. The study demonstrated that patients affected by colorectal58 had a different selective VOC spatter with respect to healthy controls. 15 on 58 identified VOCs exhibited statistically different abundances between cancer patients group and control group, and were then considered eligible for discrimination. The application of the probabilistic neural network model to the 15 VOCs showed a discriminant performance with a sensitivity of 86% and a specificity of 83%. Finally, the application of the predictive model to the blind samples allowed discrimination between the two groups with a relatively high accuracy of 76%. As regards malignant pleural mesothelioma, we know that its management is considered today very controversial due to the long latency period between the exposure and the diagnosis, and due to specific symptoms appearing only at an advanced stage. Our first study on this disease is dated 2010. However, here are reported and discussed our latest results, including in a publication on Cancer's Journal on 2020. A cross-sectional and case control study was carried out to identify a distinct mesothelioma-related disease profile. To this purpose, a data mining approach able to discriminate between patients affected by mesothelioma and healthy controls was developed and validated. A total number of 39 individuals were recruited, 14 with mesothelioma, 20 healthy controls, and 5 asbesto-exposed subjects due to the high risk of being affected by mesothelioma. Exhaled breath collection was standardized for all the involved subjects, and the applied methodological approach has been overall improved by the implementation of innovative technologies ensuring more efficient disease collection, greater retention and resolution of the highly volatile compounds, and the management of humidity-related issues. 3 different machine learning algorithms were applied to perform the classification, and 10 VOCs were identified as potentially diagnostics. For each compound, the metabolic process has been also speculated. For instance, the high levels of oxygenated organic compounds were strictly related to the altered oxidative stress and increased reactive oxygen species concentration. Moreover, 5 breath samples collected by asbestos-exposed volunteers without symptoms were processed as blind samples to evaluate the performance of the proposed model. A good agreement was generally found between the model output and the response from gold standard diagnostic exams, in this case, the computed tomography. As it's possible to observe from the probability score graph on the right-hand side of the slide, the blind cases classified as controls by the model were proved to be healthy subjects after more accurate diagnostic investigations, while the other 3 blind samples classified by the model as pathological cases were or affected by mesothelioma, 1 case, or by the presence of pleural plaques. The ongoing research is nowadays focused on the development and validation of automatic sampling devices for VOCs collection directly onto sorbent tubes, skipping in this way, as I mentioned before, 1 analytical step and reducing contamination services. In the last years, our research group, in collaboration with a leading enterprise in medical equipment, developed an innovative device for VOCs automatic collection named Mistral, and evaluated the performance through the comparison with the more consolidated sampling approaches and devices. At this regard, in our latest published paper, performances of innovative automatic devices for breath collection have been explored with respect to conventional sampling procedure using polymeric bags. One of the main purposes of this study was to evaluate the extent of the contamination reduction using the automatic devices. As I previously mentioned, the use of automatic sampling devices and the removal of 1 analytical step is assumed to limit the potential contamination sources and the interference of exogenous VOCs in the breath VOCs profile. Following a protocol, breath samples for 10 healthy subjects enrolled were sequentially collected inside a tabular bag using the automatic sampler Mistral, using the automatic sampler receiver. Each sampling session provided 3 breath samples and 2 ambient air samples, 1 with Mistral and the other 1 with receiver. In addition, 1 breath sample from a female patient affected by lung cancer was analyzed in order to make a preliminary exploration. Once collected, breath samples were analyzed by TD-GCMS with an optimized high-performance method. 2-way ANOVA data analysis was performed on the dataset. In detail, here are reported the main outcomes for the comparison between breath samples and ambient air. VOCs levels in breath alveolar fraction collected by Mistral and receiver resulted to be significantly different, 95% of significance, with respect to those detected in the ambient air, confirming the potentialities of both devices in contamination reduction. Moreover, an overall good agreement between the 2 automatic sampling devices was found. Specific target VOCs were indeed more abundant in the lower airways breath samples collected by Mistral and receiver, than in samples collected by polymeric bags. To conclude, what could be a future challenge in breath analysis research field? It could be the development of a new idea of prevention based on successive levels of diagnosis. The first level of screening could be based on the fast response of sensor devices, leading to investigate only unclear clinical cases. These cases could be defined by breath analysis applying gold standard analytical techniques, TDGC, MS analysis, and only after these two levels, expensive and invasive diagnostic tests, such as biopsies and colonoscopies, will be used. In conclusion, the chemical characterization of volatile organic compounds in human breath and the identification of characteristic metabolites is today recognized as a promising methodological approach for the early detection and follow-up of oncologic diseases. Despite the promising outcomes obtained by the scientific community over the last decades, breath analysis is still far from being applied as a routine clinical technique due to the lack of standardized procedure for sampling and analysis. More and more promising and reliable outcomes for the early diagnosis of oncologic diseases have been also obtained over the years by University of Bari Research Group in collaboration with oncology research institutions and leading technology enterprises in the field, as a result of increased knowledge and technology advancements. But other challenges for the future are waiting for us, and we are working to be ready for this. Thanks for your attention.